Hello, everyone. I'm Bob Avery, and I'd like to start the GA panel. We'll introduce our distinguished panel of discussants after these 6 quick, rapid-fire presentations on geographic atrophy. I'd like to invite David Esposito from O&L Therapeutics to the podium to give us a talk on his company. Thank you.
Thank you very much. Is there a clicker up around? Either way. All right. Hopefully just be able to advance the slides. Thank you very much for the invite at Eyecelerator. Give an update on O&L Therapeutics. I'm David Esposito, the CEO. We're based in Ann Arbor, Michigan, outside the University of Michigan, where our technology was discovered in the research labs from our founder, Dr. David Zaks, who's a retina specialist. Next slide. All right. Over the last decade or so, we've been focused on building a platform of neuroprotection. And more recently, over the last few years, we've taken our lead compound, ONL-1204, through the clinic in 3 unique programs. From the top, starting in geographic atrophy, ONL-1204, and the data I'll show shortly, demonstrates slowing lesion growth at a dosing interval of every 3 and 6 months, we think is very well differentiated in the marketplace. Next up, in open-angle glaucoma, ONL-1204 is beginning to demonstrate structure and function correlation in less than 12 months and the potential for an IOP-independent treatment of neuroprotection. And then lastly, with regards to, if you go back that slide, I think, I'm not sure if we don't have the clicker up here, but that'd be great. With retinal detachment, we're looking forward to top-line results in spring of 2024. So over the last few years, we've made steady progress across the clinic. And in the next slide, the foundation for the technology is a very unique mechanism of action focused on blocking the FAS receptor. FAS is an upstream activator of cell death and inflammation. By targeting FAS, we can protect retinal cells across a number of death pathways and also quiet the inflammatory response that ultimately accelerates cell death and tissue damage. Over the course of our clinical programs, we've now demonstrated ONL-1204 as a small peptide delivered via IVT injection is blocking the FAS receptor and has biological activity in those 3 unique disease states. Next slide. So over the last number of years, we've been reaching out for venture capital investment and made steady progress on those investment commitments across those 3 programs that I mentioned: retinal detachment, geographic atrophy, and open-angle glaucoma. We've demonstrated the unique importance of the FAS receptor and biological activity on that receptor with 1204. And we're here today to look forward to a registrational study for GA, and we're looking for investor capital to be able to move that program forward. Next slide, I just wanted to highlight our clinical program in GA. What's outlined is our phase 1b study that we've been executing in Australia and New Zealand. The study's just about wrapping up. There's 2 components of this study. I'll highlight data from both of those. The first component was an open-label dose escalation arm, looking at a single injection of ONL-1204 over 6 months. And then the second component is observing patients in a natural history component and then moving to randomization with 2 injections every 90 days. And I'd like to present that data now. The next slide. This data represents those 6 patients in component 1 of an open-label dose escalation. What you'll see is 6 patients with ONL-1204 at a single injection over 6 months, demonstrating slowing lesion growth on average over 40% compared to untreated eyes. We think 6-month durability in showing this kind of lesion growth slowing is significant in the marketplace. This was component 1 of the study. The next slide highlights component 2 of the study. So this is observation for 6 months natural history, and then what's depicted is a 6-month efficacy endpoint with 2 injections dosed every 90 days to show, again, every treated eye with ONL-1204 is slowing lesion growth compared to the untreated eyes. We think that positions us very well to move into a registrational study for GA. And on the next slide, where we're proposed to do is we look into 2024. We will be delivering top-line results in our first phase 2 study in retinal detachment. We hope to begin a registrational study in geographic atrophy and preparing to move into a phase 2 study in open-angle glaucoma. I appreciate the time and attention. Look forward to the questions from the panel. Thank you very much.
Thank you so much, David. And we're going to move on to Douglas Love, who is President and CEO of Annexon Biosciences.
Good morning, everyone. Thank you, Eyecelerator. We appreciate the opportunity to provide an overview of the Archer Phase 2 study in geographic atrophy. I'll be making forward-looking statements as part of this talk. At Annexon, we are solely focused on blocking classical complement inflammation where it starts on diseased tissue. In geographic atrophy, that's on photoreceptor cells and synapses, which to preserve function or vision in patients who are vulnerable there. We've demonstrated preclinically that we can protect photoreceptor cells and their function, and that is the basis for the findings in which we've demonstrated in our Archer Phase 2 study. There, we demonstrated the first clinical significant dose time dependent preservation of vision on BCVA 15-letter loss. That demonstration is supported by multiple lines of evidence, including 12-month treatment data, fellow eye data, and off-treatment data. And as a result, we are the first program in geographic atrophy to receive EMA Prime designation. We are now actively planning for a global Phase 3 program. I'm going to whip through this pretty quickly, so stay with me. Annexon's founding was founded based on the discoveries of our founder, Ben Barris, on the role of C1q in the classical pathway in driving neurodegeneration in a host of diseases. Importantly, blocking C1q has been shown to preserve or to protect against neurodegeneration in diseases of both the brain and the eye, and you see a list of those here. Most of these studies have been done in various labs around the globe, so we like the consistency of the underlying approach. Looking at geographic atrophy more specifically, the loss of photoreceptor cells and synapses and their degeneration occurs prior to RPE loss in GA. In the top panel here, this is a histologic section of the retina of a patient without geographic atrophy. Note that in the blue and the red, the photoreceptor cells and the synapses, as well as the RPE cells in fluorescent green at the bottom, all are fully intact across this section of the retina. However, in the lower panel, you see, this is a patient with geographic atrophy, you see the degeneration of photoreceptor cells and synapses moving from left to right across the retina long before you lose RPE cells. Of course, photoreceptor cells and synapses is where vision is housed, and our approach in blocking C1q to protect these photoreceptor cells and synapses occurs upstream of RPE loss. As a result, we would expect to see an impact on function before seeing an impact on the anatomical biomarker. The Archer Phase 2 study is a 270-patient study that included foveal and non-foveal patients. We also allowed patients with CNV in the fellow eye, and it was randomized 1:1:1 between sham, monthly, and every other month with a 5 mg dose of ANX007. There's a 12-month study with 6-month off treatment. With regard to the primary endpoint of fundus autofluorescence or slowing of reduction of lesion growth, we demonstrated a trend there with ANX007, but it was not statistically significant on this endpoint. Importantly, with longer dosing in this study, we were encouraged to see that the impact in the second 6 months on lesion growth was much larger than what we saw in the first 6 months, suggesting that with longer treatment, we will see a more durable and powerful impact on this surrogate biomarker for vision. As it relates to vision more specifically, we were quite encouraged by ANX007's statistically dose-dependent impact on the loss of vision as measured by BCVA 15-letter loss. Of course, BCVA 15-letter loss is a well-established endpoint for visual acuity, and we had pre-specified this as a secondary endpoint in the study. We also did a time-to-event analysis, and there, too, we saw a dose-dependent impact with ANX007 on BCVA 15-letter loss, showing a 72% reduction in risk in the every month group. Importantly, and consistently with the impact on fundus autofluorescent, we saw an increasing impact on the loss of a preservation of the loss of vision in the second 6 months of the study, really speaking to the potential for a durable outcome with this approach over time. Our second line of evidence relates to an analysis called a fellow eye analysis in patients who had bilateral GA. Importantly, there was no difference in the sham group between patients in the fellow eye and the sham eye. However, when you look at the treatment arms, you see a 74% reduction in the every month group in preservation of vision and 47% in the every other month. Quite encouraging from our perspective. And then finally, third line of evidence is an off-treatment analysis following the preservation of vision demonstrated in the 12-month period of the study. Once we withdrew drug, we began to see that treatment effect go away, and the sham lines, or the lines off treatment, began to mimic sham. So quite encouraging from that perspective. This is preliminary data. The full data set will be presented tomorrow at the late breaker section by Dr. David Lally at 4 p.m., and we invite you to attend that. From a safety perspective, Annexon 07 was generally well tolerated in the study. Indeed, there were similar rates of CMV in the sham every month and every other month arm. There were 3 cases of endophthalmitis, all related to procedure. There were no cases of retinal vasculitis and no cases of ischemic optic neuropathy. And so in close, we're really encouraged by this differentiated complement approach, preserving functioning cells, photoreceptor cells, and synapses for functional or vision preservation, and we're actively pursuing a global phase 3 program. Thank you for your time.
Thank you very much. I'd like to invite Brian Culley from Lineage Cell Therapeutics to give the next talk.
Thank you very much, and good morning, everyone. You're all, of course, aware that there has been a revolution in oncology, and that revolution has been driven by cell therapy. Cell therapy is now one of the 5 pillars of oncology, alongside chemotherapy, surgery, and others. What you may not be aware is that a similar revolution is poised to occur in other indications, including ophthalmology, and in particular, the application of dry AMD with GA. The reason for that, I believe, is because by the time the RPE and the photoreceptors are dying off, there is far more than just one pathway which has become dysfunctional or some group of pathways. Perhaps the only approach to really stop this disease is by replacing the entire cell to try to regain the original function. I'm going to show you some data. Some of that data is going to be completely objective. It is immune to patient effort, and it is data for which it is impossible to occur naturally. Human beings cannot regenerate their retinal tissue, and so it is very evocative to think about what an event rate of 0 on a control arm could mean compared to a therapy such as the one that we're working to develop at Lineage. Before I show you that, I am required to tell you that if you're considering investing in Lineage Cell Therapeutics, please be aware of all the risks of doing so, and you can read that all on our filings as we are a public company. Thank you. All right, so what do? We manufacture mature RPE cells. These are differentiated RPE cells. They are grown on microcarriers. The batches are manufactured in a bioreactor. Our current production scale in our labs is 2,500 clinical courses. It's just a 3-liter bioreactor. It is substantially a closed system. We have excellent control over our process. And please keep in mind that these are not undifferentiated stem cells. In fact, we cannot detect undifferentiated stem cells in our clinical material. These are differentiated RPE cells, and these are cells which are manufactured to provide extensive clinical outcomes. Let me show you what we have demonstrated. The punchline here is what we're calling structural improvement. Structural improvement is not a term of the art. It has to be defined in different ways. The data that I'm showing you has been assessed by 4 groups independently, including Doheny, including our partners at Roche and Genentech. And in fact, this is Roche and Genentech's current version of defining their threshold for structural improvement. It's a reduction in subsistence, a reappearance of ELM, and increased hyperreflectivity. when we see that, you can see these striking improvements in the anatomical structure of the retina, which are occurring in as early as a few weeks, in this case, this patient by 3 months. Again, this never happens spontaneously. Following this patient out for longer now, the same patient out for 24 months, you can see a durable change to the anatomical structure. In this case, the patient has an RPE gain, not less loss. This is an RPE gain, an ELM gain at 24 months. When you go out even farther, you can start to bring in more subjective assessments such as BCVA and ask questions such as, wouldn't I expect a patient like this after 3 years to have approximately 2 lines of BCVA vision loss rather than being at baseline as she is at year 3 and year 4 compared to her contralateral eye, which has lost 30 to 40 letters. I recognize that this is BCVA and it can be very subjective. This is not the only patient that has demonstrated these effects. We actually have 5 patients who have exhibited these same kind of effects, notably anatomical changes coupled with functional benefit. All 5 of these patients had BCVA above baseline at their 12-month assessments. Again, this never happens naturally. It also never happens with anti-complement therapy. If Cedric had a patient that had this kind of thing, we'd know about it, right? This doesn't happen with anti-complement therapy. This may be the only way to drive these kinds of changes. These same patients, they all had something in common. They had extensive coverage of our cells across the area of atrophy. And on average, they gained 12.8 letters of visual acuity. They had a gain in RPE. Patients that received the cells anterior to the area of injury did not exhibit these similar or comparable changes. So this is very much a transplant. This is not trophic factors leaking away and driving clinical effects. The cells have to be there. It's more like a bone marrow transplant than what you might think of from the chiropractor down the street charging $5,000 for spun down fat cells, right? That's not what we're doing here. Just briefly, I want to mention with respect to safety, these patients are not on lifetime immunosuppression. Please dispel your mind of some of the now archaic ideas around cell therapy. This is perioperative short-term immunosuppression, just 90 days. We have had no cases of cysts, no teratomas, no tumors. These patients, we have now approximately 100 person years of exposure, 0 cases of rejection of these cells. This study is now moving on from our hands to our partner Roche and Genentech. They are actively enrolling in a phase 2A study. I'm very excited about this as it may represent the future of GA therapy, and our objective is to try to erase the word irreversible from this disease. Thank you.
Thank you so much, Brian. Next up, we have Mohamed from Aviceda Therapeutics.
Alexandria and Bob, thank you, and thank you for Eyecelerator for the invite today. All right, let me see. So, this is Mohamed. I'm one of the co-founder and the CEO and the president of Aviceda. Let me just introduce Aviceda to the team here. Aviceda, we're still a private clinical stage biotech company. We are based in Cambridge in the US. We are a technology platform company using this nanoparticle to target our target biology, which is called SIGLEC. It's a glycoimmune platform. Our lead molecule called AVD104, it's being designed for patient for GA second to the AMD. As everybody knows in this room, I hope they know, there's significant unmet medical need in patients with AMD and also retinal vascular diseases. AVD104 is a highly disruptive intravitreal nanoparticle being formulated to target patients for GA to show better efficacy and better safety and a longer durability. This program is currently in a phase 2/3 called SIGLEC trial. It's have 2 parts. Part 1, I can announce, is being fully enrolled since last month. We're going to release some of the data today with a full data analysis will be released by end of this year, early next year. The second part of the study, part 2, is actively enrolling in the US, and we're going to give you some background about the design in a minute. The team, built the company with me, most of them are retina specialists. Most of them are in this room. Dr. Callanan, Dr. Kunimur, Dr. Tolentino, Dr. Hassan, and the rest of the team are executive committee members with very strong background in business and drug development. In addition to the team, we have a very strong clinical advisory board. You know these people very, very well. Let me just introduce you to the technology in a high level today. The technology called HALOS. HALOS stands for High Affinity Ligands of SIGLECs. SIGLECs, if you don't know the glycobiology, this is one of those classes we never took during the college time. but I learned about the Cedric over the last few years. Ceglics are immune resolution receptors. They actually present on all our immune cells, innate and adaptive. They need sugar, sialic acid, to interact with Ceglics to drive the immune resolution, either on or off. We built a technology based on that. We have a very strong, a large library of high affinity synthetic ligands that could ligate and be conjugated to a modular nanoparticle we have in-house. This is how we build our lead acid. We just talked about the immune oncology in a different presentation. It seems all the innovation starts from oncology and migrate to the ophthalmic space. Let me just give you some relevancy here. This revolution started, in the immune oncology space when Keytruda and the others, the ICI, the immune checkpoints inhibitor, came years back in cancer or immune oncology, like PD-1 and PD-L1 receptors. They drive the immune resolution or activation in those cells like Ceglics, the same item domain, the intracellular domain. The beauty of the Cedric receptor, they are present on all the immune cells versus the PD-1 only T cell mediated. Let me talk more about ABD104 and GA and how this is relevant to the discussion here. Most of you right now, we know AMD is two pathways, the cellular component macrophage overactivation and the humoral, the complement activation drives the immune activation and drive, the cell degeneration and the cell lysis. Right now, we are very, lucky. We have, a couple of drugs, in the market, target the complement. But as we know, it's not enough yet. A dual MOA beyond complement potentially can provide increased efficacy for GA by targeting the two pathway, the cellular and the humoral. This is a histopath, data from one of the patients with GA. If you look at the edge or the junctional zone on the boundary of GA, you'll see two things. You'll see activated microglia and macrophages, and also you see complement. No wonder. But that's why we need to target both. If we target one, it's still going to be good, but it might not be good enough for our patient. The other thing is very important to know, macrophages are the predominant cell into the leading edge of the GA lesion, and also the one produce the local C3 and complement factor in retina. So ABD104 targeted it's a dual MOA, targeted two pathway, the cellular component, by inhibiting the overactive macrophages and repolarize them to the resolution state. We need macrophages for our biological system. At the same time, not independently, not sequentially, we target also the humoral component of the disease by activating CFH to normalize and inhibit the complement overactivation, overproduction. This molecule right now in a phase 2/3 trial based in the US, the trial called Cedric. The trial was designed into two parts. I'm going to report some of the data from the first part today. The part one was a single ascending dose, open label, multi-center trial based in the US. We enrolled, 30 patients in the trial since last month. The primary objective, as usual, was safety and tolerability, looking for oxysystemic and also dose-limited toxicity. The secondary endpoint, we're very keen to look if there's any early efficacy and also some biomarker analysis we're going to report today to you. The patient was centrally involved GA, very typical advanced GA patient, and the lesion was larger than 1 on 25 millimeter square. The trial, we only injected one single injection with ABD104 in part one, in multiple dose, and each patient being followed up for 3 months. I'm very pleased as a retina specialist to see all the patients in all the cohort thus far didn't have any safety signal whatsoever. The drug was very well tolerated from the low to the highest dose we used in the trials. We didn't see any drug-related adverse or serious adverse event, either ocular or systemic, up to 3 months. Most importantly, we didn't see any dose-limited toxicity per the protocol criteria. We did look at other, adverse events of special interest, including, there is no IOP changes, no significant visual loss. We didn't see any case of retinal vasculitis up to 3 months, no CMV, no ischemic optic neuropathy, no intraocular inflammation. The study is still ongoing. The full data will be released by end of the year. The other critical thing we looked at was treatment effect. And I'm very excited to have brought this to the street for the first time. We have up to 30 patients enrolled. The data I'm presenting today is only out of 15 out of 30. What we saw for the first time in GA space, we began to see visual acuity improvement. This was PCVA using the typical ETDRS chart. We found in 12 out of 15 patients we have data from, 80% of the patients have visual acuity gain. And when we did more category analysis and we looked how many patients gained 5+ letters, and from 0 to 5, we found 40% of the patients, 6 out of 15, improved 5+ letter. It was a very clear also dose response. The higher you go, the better the visual acuity gain. We only have 3 patients in that study lost 2-3 letters from baseline. That visual acuity gain for the first time also was very
Can we summarize or move to the next slides?
Sure.
We're over time.
That visual acuity was also very consistent with the GA lesion analysis. We saw some stabilization and regression in our 2 lower cohort. This is an example of another patient, show loss or regression of the lesion with PCVE gain in study eye, and the biomarker which we collect from ex-humor show the inflammatory biomarker and the complement factors are reduced as the drug should work. Part 2 of the study is actively ongoing. It's a double-masked randomized active control study, 290 patients to be randomized among 40 sites in US, 75% extrafoveal. The primary endpoint will be a rate of change from baseline. Secondly will be change from baseline PCVA maintenance, also looking at OCT structure analysis. In summary, current complement inhibition only addressed one arm of the innate immune response and may deplete the complement factor. AVD-104, a sialic acid-coated nanoparticle, repolarize overactivated macrophages and inhibit over-amplified complement system. Part 1 and Part 2 of the SIGLEC US trial are underway and currently enrolling. And AVD-104 show no drug-related systemic or ocular safety event. Thank you so much for your attention.
Great, thank you. I'd like to introduce Cedric Francois from Apellis, please.
Thank you, Bob, and thank you so much for inviting us to present here again. I will be making some forward-looking statements. And this is our company. We have 2 drugs on the market, actually, one for a systemic indication called paroxysmal nocturnal hemoglobinuria, with a drug called Empaveli, and then, of course, Syfovre, which we are all familiar with. Here are the results that are now already a few years old, the primary endpoint readouts, and then the follow-up at 24 months in Derby and Oaks, where both with monthly and every other month injections, we showed a reduction in the lesion size growth in patients with geographic atrophy. What was really interesting and captivating to us at the time as well was that these effects over time seem to increase, where the curves widen. And we saw this both with the monthly as well as with the every other month treatments, reaching approximately 30% in the monthly arm in that segment from month 18 to 24. And then we have the GALE extension study. So after patients have been on treatment for 2 years, they go on for another 3 years of treatment. That 3-year time point will be presented at the Retina Subspecialty Day on Saturday. And what was most gratifying there to see is that, again, we see these increasing effects over time continuing to mature. Now, what we have here is a theoretical sham, because the patients are, of course, switching towards active, but these lines continue to diverge in a way that we had expected. What is also important is that patients that went on active show the same response pattern as the patients did at the beginning of Derby and Oaks as well. We saw this effect both for non-subfoveal as well as for subfoveal patients. And as you can see from this graph, what is interesting is that by the time you get towards the end of the 2-year period, both non-subfoveal and subfoveal patients seem to respond with similar reductions in lesion size, again in that range of 30 to 40%. Now you could say, okay, the sham is no longer there. How do we know for sure that this is a real effect? Well, the best way, in my opinion, to do that is to look at the effect vis-a-vis the fellow eye. And these are, of course, large sets of patients that we look at. And what you can see here is that, again, both with monthly as well as with every other month injections, we see these increasing effects reaching around 30% in the treated eye compared to the untreated fellow eye. Now, of course, function is something that is very important that we try to correlate. And the reason why we look at lesion size growth in geographic atrophy is because it is so hard to evaluate function in the back of the eye. The two most objective ways to do that are probably visual acuity and microperimetry. Now, visual acuity is complicated in the sense that it only really looks at central vision. I mean, you can look through a straw and come out with 20/20 vision, but of course, functionally, you would be significantly debilitated. But it's an important measure. With microperimetry, the issue is that it's a difficult test to do. So what we have here is 68 dots in the back of the eye that are sequentially tested in a patient with increasing light intensities, and where the patient then says, I can see this light or I cannot, dot by dot. So it's a really good measure to get a broader picture of what goes on in the retina with the limitations that I outlined. Now, when you look at all of these points in the context of geographic atrophy, it can be complex because these points that are sitting inside of a pre-existing scotoma are not going to see, and that's not going to change. Now, if you go far away from the lesion, these points will see well and also are not going to be affected because with the natural growth, those points would not be implicated anyway. So as we looked at microperimetry, we thought it was important to take two measurements under separate analysis. The first one is when you look at the border around the lesion of geographic atrophy, that is a perfect place to really get an assessment on whether there's an impact on function or not. And what we saw there was that over the course of 24 months in Derby and Oaks, that we saw, similarly to the lesion size growth, an increasing effect over time. Now, these are, of course, post hoc analyses, but it's intriguing how both on scotomatous points and threshold sensitivity, we saw that. Now, when you go to the 4 central points in the back of the microperimetry analysis, what we see there is a clear correlation with visual acuity. We saw that at points at month 24, both with the 4 central scotomatous points, as well as when we did this with the 16 central points in that central vision. And what you have here is essentially patients that still have non-subfoveal lesions. They still have vision left in the center of their vision, and then the question whether drug can reduce the progression to that. So these are post-hoc analyses, but very exciting and create a clear correlation between lesion size growth and function. Also, on the photoreceptors then, with OCT, you can segment the photoreceptors from the retinal pigmented epithelial cells. And what we see there, and it's been mentioned a couple of times, what is fascinating is that photoreceptors seem to benefit completely from complement inhibition pretty much from day one. Now, they can sit outside further away from the border of geographic atrophy, and when the RPE cells then catch up with the photoreceptor cells, then they start dying in conjunction. So much more to be studied there, but a very fascinating observation. So we look forward to continuing to work with all of you. I want to express my gratitude to everyone here present that has helped us get to this point. We expect a decision from EMA early next year and the other geographies following there as well. These are the presentations that we will have in the next couple of days, and I want to thank you for your attention.
Thank you, Cedric. The last presentation before the panel discussion is going to be by Erin Henry from Iveric Bio.
Thank you to the moderators for allowing us to provide an update on how we're innovating in geographic atrophy at Iveric Bio, an Astellas company. I will be making forward-looking statements per this disclaimer. Our company was founded in 2007 and rebranded Iveric Bio in 2019. In July of 2023, we were acquired by Astellas Pharmaceuticals, and in early August, we received FDA approval of avacincaptad pegol as an FDA-approved treatment for GA secondary to AMD. I'd like to take a moment to talk about the specific design of avacincaptad pegol. It is a pegylated RNA aptamer. It's small in physical size and designed to be synthetic in production without any biological components. It targets the complement cascade at the level of C5, thus preventing activation of the inflammasome as well as formation of the membrane attack complex. We believe it is these activation steps that contribute to the cell death associated with the progression of geographic atrophy. Importantly, we also maintain the upstream components of the complement pathway, particularly the anti-inflammatory components through C3a. Avastin-capsid pegol was evaluated in 2 pivotal trial programs, the GATHER1 and GATHER2 studies, and we saw a statistically significant reduction in the patients treated with Avastin-capsid pegol on their GA growth compared to sham. This is over 1 year and was the basis of our FDA approval. Looking at the most frequent ocular treatment emergent adverse events in the study eye, we see that most of these reported are those that we would attribute to the injection procedure. However, I've highlighted the increased number of choroidal neovascularizations and the increase in intraocular pressure, which was transient and temporally related to the 100 microliter injection procedure. Importantly, we spend a lot of time looking at adverse events of special interest. This is of high interest with new therapeutics in this space, and I want to show you the 12-month results for both GATHER1 and GATHER2, which have been previously presented. We see that for intraocular inflammation, we have a single mild case of vitreous cells that resolve without any treatment. We don't have any cases of endophthalmitis or ischemic optic neuropathy, and there were no cases of retinal vasculitis in this program. We've seen longer-term follow-up for GATHER1 that is published, and you will see shortly the longer-term follow-up for GATHER2 when we present the second-year results on Saturday. As a reminder of the GATHER2 study design in anticipation of the GATHER2 results that we will share at Retina Subspecialty Day, I want to just remind you that this trial protocol is different than GATHER1. It's also slightly different than what you may have seen of other complement inhibitor programs to date. In the first year of this global prospective study, patients received monthly ACP or sham monthly. They had the primary analysis at 1 year, which I just shared with you. For those completing 12 months in the ACP arm, they were re-randomized to either remain on monthly dosing or move into every other month dosing. And then if you were in the sham arm, you remained in the sham arm through the second year of the study. So what we're evaluating in the 2-year objectives is the efficacy and the safety of the monthly dosed arm over 2 years, or the patients that moved into the every other month arm over 2 years. And that's what we will be presenting shortly. But I think you may have seen that we presented a press release in mid-September, and I just want to highlight that in that press release, we announced that the primary analysis of the second-year results, the monthly treated arm over 2 years, statistically significantly reduced GA growth relative to sham. We also said in this press release that the every other month dosing arm had a treatment benefit that was similar to the monthly treated arm. And importantly, we're seeing consistency in our safety profile over the second year of the study. This is so important, and we're happy to report that we don't see any new safety signals emerging. I welcome you to see the full, detailed results being presented by Dr. Arshad Kanani on Saturday morning of Retina Subspecialty Day at 10:23. Thank you so much.
Thank you, Erin. Can we pull up the polling question now? And while that question is being pulled up and voted on, I'd like the panel to introduce itself. I think, why don't you start? And then we'll go this way.
Fantastic. So wonderful presentations. It's such a pleasure to be here. My name is Aleksandra Rachitskaya. I'm practicing retina specialist at Culley Institute Cleveland Clinic.
So please vote for this polling question. And Cedric, go ahead and introduce yourself.
Yes. Oops. Cedric Francois, CEO of Apellis.
Wiley Chambers, Director of the Division of Ophthalmology, Center for Drug Evaluation and Research, FDA. Namrata Saroj, independent consultant in Retina.
Erin Henry, Vice President of Product Strategy and Innovation, and the Avastin Captive Pegol Asset Lead for the Global Development Program.
Yeah, hi, I'm Dan Lyons. I'm a portfolio manager at Janus Henderson Investors. Thanks so much for having me.
Thank you. Well, we're getting the results. It looks like the ONL fast inhibitor is winning right now. I don't know if it's quite done yet. We got, I think, almost 100 people responding. With respect to that drug, it's very interesting that it's working as a neuroprotectant in 3 different areas. Does anyone from the panel have any thoughts on how this is so pervasive in so many different diseases? Well, I would postulate that neuroprotection is something that's really important for lots of different diseases, and if we can stop these early pathways with a single agent, it may work for multiple different components of our ophthalmic diseases. Down those lines, I noticed, Cedric, your slide was very interesting at the end of your presentation on the photoreceptor preservation early on in the treatment. We always had this sort of dichotomy with, we had vision separate from anatomy with GA treatment. And it may be that it has to do with this sort of protection of the photoreceptors and the ring around the geographic atrophy. Maybe it's this neuroprotection or saving of the photoreceptors that happens separately from spread of the RPE loss. And that may be what's happening in Annexon too, where they have a visual improvement when they're not even really seeing much improvement in the anatomy early on. I'm curious as to everyone's thoughts about what is going on with the difference between the vision and the anatomy. Any comments?
For me, this is one of the most fascinating observations that we've made, actually. It's difficult when I try to explain it to people that are not familiar with the retina. I compare it to a lawn with grass, where you have a hole in the ground, and then an area around it where you have no grass but ground, and then beyond that the grass on it, and where essentially, from what we see in these analyses, and this is done in collaboration with Ursula Schmidt-Erfurt. This fascinating observation that the grass kind of stops dying immediately, and then when the ground catches up with the grass, they both start dying together. And probably, the photoreceptor cells then die because they're no longer supported. But I think it's a really interesting observation. For me, it creates hope that if you can find kind of the next button in the RPE to get to that same stoppage as you do in the photoreceptor cells, you may have a very effective and immediate therapy in geographic atrophy. And to your point, I think in all of these experimental studies, it's interesting to look at.
Well, yeah, so I just want to add to what Cedric said. I think we're just in the beginning of the spectrum of learning about GA, and especially what we're testing with imaging as well as with vision, we might not have the right tools. So as we're making progress in drug development, simultaneously this progress being made in understanding assessments, and I think we're going to learn more about the correlations as we move forward.
So I think it's a fantastic point, Namrata, that you bring up. When we listen to this fantastic, excellent presentations, and I keep thinking about outcomes, right? It's so hard to design these clinical trials, and both Apellis and Iveric, you guys relied on the fundus autofluorescence as the primary outcome. But from your Cedric's talk, and when we look at RP cells, maybe we should be looking at OCT more and analyzing that. Maybe I'll ask Wiley. Wiley, what do you think about looking at the imaging outcomes in GA, and particularly role of OCT?
So I care because I think patients care more about what they see, not what an image shows up. So the first thing we would like to see from the FDA perspective is whether patients are either seeing better or losing vision slower. And I think, as you just, I agree with the other comments you've heard. I don't think we have the technology at the moment to monitor visual fields as well as at the same level of where we're having the action take place. And I think when we get a better handle on visual field loss, we'll have a better understanding of what's happening.
Would something like the ellipsoid zone, which I guess for instance, for MACTEL might be an approvable endpoint, what would need to be done to sort of validate either photoreceptor loss, as was measured by Cedric, or ellipsoid zone loss as being an endpoint for geographic atrophy? What sort of validation would you require?
So I don't think there's any controversy that if you don't have a photoreceptor, you don't see at that particular point and spot. If I'm wrong about that, I'd love to hear people tell me why. So that's why we accept preventing photoreceptor loss. That's different than adding photoreceptors to it, because if you add photoreceptors to the retina, you have to not only make sure you've added the photoreceptor, but it needs to work. But as far as loss, once the photoreceptor is gone, we think that's something that should be prevented. So whether you measure that by autofluorescence or by EZ zone, we don't particularly care. What we care about is whether the photoreceptors are there or not. Beyond that, so far we're still looking at visual function.
Cedric, do you think microperimetry plays a role? I mean, it is an assessment of visual function, but how easy it is to do in those patients?
I think where we're going will be microperimetry, but not with the patient responding, but with automated responses, either based on some kind of electrophysiological determination that the signal has occurred, or whether it's a pupil response, or whether there are a variety of ways to take basically not require the patient to be doing the responding for you. But I think ultimately we will come up with visual function that is in that, or visual fields that are representative of that.
I have one more question for you, and then we'll move on to the rest of the panel. Sorry, guys. You know, ONL got a lot of data out of their phase 1 study by looking at the run-in, what the rate of atrophy progression was before they were treated, and then also comparing it to the fellow eye. Is there any way in a more registration trial, fellow eye could be a portion, or a run-in period like that could be a control?
Yeah, I am not anticipating that we'll use fellow eye. Okay. If the endpoint was 10 years down the road, I think fellow eye is perfectly fine. But for a 1 to 3 or 4 year period of time, eyes are not directly correlated to be that, at least from my perspective, that you can use the fellow eye.
And will these companies going forward, will they have to use a non-inferiority style trial against one of the approved drugs for approval?
It's actually worse than that. You have to use a superiority trial.
Yes, I'm sorry. There's a whole session on this later. Stay tuned.
We don't have a non-inferiority margin.
Okay. Yes, I get it.
So switching gears a little bit, you know, Erin, this is for you. You've presented beautifully the results of the clinical trial, and we'll hear more on Saturday. But now we have real world, right? The drug is in the real world. So can you comment on the experience in real world, and maybe comment on safety as well?
Absolutely. So Eyecelerator V was approved in early August and commercially available beginning in September. As you saw from the clinical trial experience to date, we feel that the drug is well tolerated, and we continue to see signs of that in real-world utilization. I think there is worth noting that we have a single post-marketing safety report of a vasculitis case, and this is a really complicated case. It was an off-label use of our medicine in a 43-year-old Stargardt's patient. The first eye of this patient was treated with Syfovre, the left eye. 4 days later, the right eye was treated with Eyecelerator V. The Syfovre eye developed vasculitis symptoms, and then 2 days later, the Eyecelerator V eye developed symptoms. Now, this is an ongoing case, and we continue to update the FDA and other health authorities as we understand it better. But I think out of an abundance of caution, the clinical learning that I have on this is that We need to be able to appreciate the safety of these products in the real world rigorously through the spontaneous as well as company efforts to report and be transparent. And then if you are using Syfovre, we would ask that you give that eye a sufficient period of time before introducing another product into the same eye or into the other eye, just out of an abundance of caution. Thank you.
Cedric, in follow-up, what do we think the mechanism is of these severe retinal vasculitis cases, almost the HORV or hemorrhagic occlusive retinal vasculitis we saw from vancomycin, intracameral vancomycin? What do you think is actually going on in these cases? Any clue, especially now that we know it can happen with both agents, at least in this one unusual case?
Yeah, so it's tempting to speculate, right? I mean, yes, and there are and there are many hypotheses that we are exploring. We are working very hard at Apellis to kind of look into what potential contributing elements could be. But what's really important is that the instance of these events is extremely rare. That is a very good thing, both on a per injection basis as well as on a per patient basis. So we have in the real world, north of 100,000 injections now, with a rate of about 0.01% in these vasculitis cases. We also have approximately 50,000 patients that have been injected. And if you take vasculitis leading to severe vision loss, then there you're also talking about approximately 0.01%. So against that background, the drug is being prescribed. We're having a lot of injections, which allows us to continue to evaluate this. And again, I mean, there's immunology. There is, of course, the needles that we changed. I mean, there's multiple factors, but for any of those to teach us something, we're going to need more time. So that's the...
It's interesting, we've heard about all the cases that were presented at ASRS. And do you have any updates since then for us on cases that you guys have seen?
Yeah, so, this subject that Erin was talking about was a subject that was from the second half of September. But in patients with geographic atrophy, our last case was from September 1st, right? So we are doing somewhere in the neighborhood of 4,000 to 5,000 injections per week now. So, we keep tracking this. We're very happy that there's good reporting. We are in close communication with the SRS REST committee on this as well. And again, it will take more time to evaluate this, but that's where we currently stand.
Great. I'd like to change subjects a little bit and go to Dr. Lyons. And, as a manager of a fund that focuses on biotech, I'd just like to get your perspective on what you look for in these early companies in innovating in a new field of therapy. What is it that you want to invest in these companies, and what have you learned from the sort of rollout of both of these companies in this field? What is it you look for to pour money into?
Yeah, thanks. So we've been investors in several of the companies in the GA space, and our process is basically first to focus on the clinical risk. 90% of drugs that are coming into development are going to fail, and we want to make sure that we're investing behind the ones that are going to make it. And thankfully, there's several examples on stage here that have. So the complement mechanism, there's good genetic association, so that made us comfortable investing behind that mechanism, and then also, thanks to the FDA, there was a clear clinical path, forward for these programs, measuring GA lesion growth. So having a clearly defined endpoint was critical for us in gaining confidence that they could make it to market. And then the second part of our analysis is always around the commercial opportunity, and there we reached out to the retinal community, and through surveys and attendance at meetings like this one, we got a high degree of confidence that this was a really important unmet need, that that the community was going to embrace these new therapies, and that we'd seen that in the early uptake of the products in the market. So one thing we like to do is triangulate on other markets like the wet AMD market, and it was very clear that there were similar numbers of patients already in practices with with GA that were in need of therapies, and there was going to be a strong uptake of when there were new products available, which we've seen so far. So we're hoping to see even more innovation in the space, and it's great to see the companies presenting today as well.
Great. I would just ask, what about the market size do you think, and this could go to the companies here as well, with this increasing uptake that Cedric just mentioned, 5,000 cases a week being injected, even with some safety concern, is the market bigger than we initially thought, or are we just having more excitement about it because this is an untreatable disease for all these years? Or what are your thoughts on the size of the market? Okay, Erin?
I'm happy to take that. So the latest numbers that we've seen from the American Academy of Ophthalmology is that geographic atrophy is present in over 8 million patients worldwide. I think the challenge of a disease that has no treatment is that you didn't have an opportunity for people to be in front of retina specialists seeking treatment until the advent of these recent approvals. And so we'll learn, I think, as there is more education that there are treatments available, as retina specialists also are able to educate their referring pathways, I think we'll have a much better idea. But from what we've seen in the clinical trials, there is an unmet need. Patients desperately want treatment options for this disease, and so I expect that we will see continued interest and better epidemiology with more time.
Yeah, just to add to that, I think we saw that with wet AMD and diabetic retinopathy as well, where once the treatments became available, the early diagnosis played a really big role in increasing the prevalence, and I think we're going to start seeing that here as well.
Namrata, I have a question for you. You consult with so many of these early stage companies. I don't think with Lineage, but I was impressed with these improvements in the structure. It sort of surprised me that you could have so much improvement in structure when the retina had already been atrophied. Do you have any comment on that? Are you also
Yeah, I completely agree with you. I think we're seeing some really great innovation coming in with these improvements that we maybe didn't expect with sort of the first generation treatment options. But that's the beauty of drug development. We need to move forward. And I really do believe, I look at everything in spectrums and I go, we're just in the beginning of understanding GA. Now that we have treatment options, we'll understand the disease even better. And we're going to continue to see more sort of this anatomic vision correlations as well as hopefully improvement. And Bob, you and I talk about this a lot. The goal really is earlier intervention into intermediate AMD so we actually avoid GA, and I think we're making progress there.
I think that's a very good point. Although the pathway is a little hard with intermediate GA, but we can talk about that in another session on pathways and endpoints. I think we're pretty much at time. Do you want to?
Yeah, I think this is a great discussion, and it's there's so many points that have been touched upon, and potentially identified in patients earlier, treating them earlier. Hopefully, we'll have treatments that not only reduce the growth, but stop the growth and reverse some of the damage of geographic atrophy. So I think it's a very exciting field, and I would like to thank all of you for joining us for this great discussion. And thanks to all of you for attending.
Thank you very much, and stay here for the winning pitch up next. Thank you.